Okay, this is a recorded lecture to accompany Chapter 16 in the OpenStax textbook. It's our introduction to disease and epidemiology. Epidemiology is just the study of disease in populations. And, no matter how many times I remind students that microbiology is more than the study of microbes that cause disease, that there are good things microbes do and that they are important in the environment, and 98% of them don't make us sick. The first thing I think of when I think of microbiology is disease. And so we're going to talk about the microbes that are associated with people. And then we're going to talk about some important history of this and to provide a little bit of background for disease and epidemiology. Right. So we have not, as I said, not all microbes are bad. Lots of them are found on our skin, in our mouth, in our digestive tract, and other places, and they're okay. Some of them are even good for us. Some of them are, they live off us without harming us. That's a commensalism. Others actually benefit us. They keep bad organisms out. They maybe improve our skin or our digestive tract. But inevitably, some of them benefit at our expense. That's parasitism. The normal flora includes some organisms that are most times they're okay, but sometimes they may be bad and they may be opportunistic pathogens. They get us when our defenses are down. Pathology is the study of disease, and it includes those organisms that are mostly good but sometimes bad. When we study disease, we want to find the cause. That's the etiology. We want to identify those organisms that can cause disease. Those are the pathogens. And we want to know how they colonize our body. How do they start an infection? And how can we, how do they get to us? And are there things that we can do to prevent disease? Sometimes it's to stop and kill those organisms. Other times it's just to change the relationship a little bit. Okay, so the normal microbiota are the organisms that are on us. Most of those are our symbionts. They're there all the time. They're our normal flora. Some are there for only a short time. They're transient. Many of those normal flora are they protect us. They antagonize organisms that otherwise might cause us problems, and so that's really important. This picture here shows the sites that are sometimes have a normal flora and that are important. The study of disease really kind of began with some pioneers that I've already kind of talked about. Jon Snow is mentioned in your textbook. He's considered the founder of the study of disease in populations, epidemiology. And he didn't identify the cause of disease, but when there was an outbreak of cholera, he started looking at how cholera occurred in neighborhoods, and he started mapping cases. And he found that cases clustered on the map. And he figured out that cholera, this terrible disease, clustered in neighborhoods and often around water sources. And so he drew this map, he provided evidence, and he didn't know what caused cholera, but he was pretty sure it came from water. And so his famous conclusion was to take the handle off the pump in the most infected area, and that stopped an outbreak. A similar person that you might have heard of is Florence Nightingale, another English person, English woman. And she was moved by pity for her countrymen that were off fighting a foreign war. And so she went to take care of the injured and sick soldiers to Crimea, okay, which is in the news now. She went there. And like Jon Snow, she was an educated person that started to think about disease and what caused disease. And so she started looking at the things that made soldiers sick and their causes of death. And she looked at that for each month of the year, and she identified what caused those deaths. And she found that Although wounds were a serious problem, they were not as big of a problem as disease. And so she started to, like many scientists of this time, by keeping track of things, she was able to greatly improve the conditions. And so an important advance. Okay, again, Lister, who I talked about, a little bit later than these people, he was concerned about the infection rate and the fatalities after surgery. And he thought, I'm a pretty skilled surgeon. Some of my colleagues are good, but people still die. And maybe this is caused by microbes. And so he had an effective disinfectant that he thought, well, I can try and use this to keep these wounds, to keep contamination out of these wounds. And so he used carbolic acid. We now call that phenol. And he used that to clean his surgical instruments, to wipe the patient's skin. And he actually, he worked in a mist of this to make sure the surgical site remained free of infection. Okay, so all these people, by collecting data, they were really studying disease in populations, not just an individual case or a clinical case, but in how these diseases were spread and what was the likely cause. And they're all focusing on patterns of disease occurrence, patterns of transmission, and they're trying things to try and disrupt that. Okay, and often they find that diseases sometimes aren't transmitted from one person to another. You can't really affect that. They're non-communicable. But other times they can be. And sometimes they're easily spread. They're contagious. And so that's really important to identify this. And that's the beginning of that study is the beginning of being able to take effective measures. Okay, and so we're going to talk about disease, not in a clinical way so much, although I will mention how the patients are affected, but also in a population sense. And in a population sense, we're talking about epidemiology and the goals of epidemiology, which are really identify the cause, find the source in the environment, the reservoir, and find out how people come into contact with that source, the route of transmission. If you know those three things, you can try different methods to control infections. And so here I have a picture of Typhoid Mary, a famous American, not for a good reason famous, but for another reason. And really they were starting to apply these concepts of systematic study and looking for patterns all over the world. And in the late 1800s, early 1900s, typhoid fever was more and more disappearing. This is really a disease caused by salmonella infections that are caused by contaminated food and water. But the symptoms were well known, and it wasn't really known the cause, but it was known, okay, it's contaminated food or water. And so Typhoid Mary was a household servant, a cook, and she was associated with 3 separate outbreaks of typhoid fever that killed wealthy people and identified each time as the likely cause. Okay, so next time we're going to go into some more details, particularly about routes of transmission, reservoirs, and different types of control methods. But this is our introduction. Start reading chapter 16. We'll have one more lecture on that.